Start.
Good afternoon, dear students. Myself, Mr. Ashutosh Upadhye. I will be talking on the topic of chemical properties of DNA. So, as we know that DNA is one of the important molecules for life. It provides the blueprint of each and every trait of an organism. So, let's see what are the different properties of DNA. So, let's begin with the components of DNA. So, DNA is having majorly three different components. The first component is the phosphate group. Second component is the ribose sugar, which is actually pentose sugar here. And the third component is nitrogenous base. Okay, there are three different types of nitrogenous base that I am going to show you now. Okay, so there are different constituents of the DNA nucleotide. First of all, when base and sugar is added, it forms the nucleoside. Okay, so you can see here the nitrogenous base adenine. When added with ribose, it forms adenosine. Similarly, when adenine is added with deoxyribose sugar, it forms deoxyadenosine. Okay. But when these base and sugar are coupled with phosphate group, this forms the DNA nucleotide. Okay. So let us see how it forms the DNA nucleotide. Okay. So if we take the sugar as deoxyribose sugar and adenine group is added to it, it forms deoxyadenine monophosphate, that is called as deoxy-AMP. Then when again extra phosphate group is added to it, it forms diphosphate, that is deoxyadenosine diphosphate. And when third phosphate group is added to it, it forms deoxyadenosine triphosphate. Okay. So this is how the composition of nucleotide between DNA happens. Now let us see what is the basic difference between DNA and RNA. You can see in this diagram the proper difference between DNA and RNA is shown to you here. Okay. This is the structure of RNA. You can see here at the second position of the ribose sugar, there is a presence of OH group. But in DNA, at the second position, the ribose sugar, OH group is absent. Okay, so here extra OH group is not there, whereas it is present in RNA. Second difference is the presence of base. Okay, so in case of DNA, the nitrogenous bases are adenine, guanine, cytosine or thymine. But in case of RNA, the thymine is replaced by uracil. Actually, uracil modification creates thymine. That is 5-methyl uracil is thymine. Okay, so this is the basic difference between DNA and RNA. So let us see what are the different components of DNA or the nitrogenous base. Okay, so the nitrogenous base majorly are of two types. First type is pyrimidine and second type is purines. You can see there are two types, pyrimidines and purines. Okay, so first let us see pyrimidines. Remember, pyrimidines are six-membered ring. Okay, it's a benzene ring type molecule where three different types of bases are available. First is thymine, second is cytosine and third is uracil. The uracil is present only in RNA and thymine is present only in DNA. Okay. Now let us see what about purines. Purines are six-membered rings which is fused with a five-membered ring. Okay. So there are of two types. First is adenine and second is guanine. Okay. So these rings are not only made up of carbon but there is nitrogen also. So therefore this is called as nitrogenous bases. Okay. So let's see what are the different nitrogenous bases that are available. So as I told you, purine is a six-membered ring fused with a five-membered ring. So it shows the structure of purine. Two purines are here. This is the basic structure of purine molecule and this is the derivative, that is adenine, formed from it. And this is the guanine that is formed from it. There is basic difference between adenine and guanine in the position of the groups that are added. You can see in adenine, amine group is additional at the 6-number carbon. Here it is present in 2-number carbon along with the double bond O oxygen at the 6-number carbon. Okay. Similarly, the parent pyrimidine molecule looks like that. It is having three different types. First is cytosine, then uracil and then thymine. All these three nitrogenous bases, that is pyrimidine, are six-membered rings. Okay. So let's see how they looks into a single strand. Okay. So when a single strand of DNA is created, it is having always a polarity of 5' to 3'. The uppermost base is having a 5' phosphate end and the lowermost base is having the 3' hydroxyl end. So it creates a polarity to the DNA, that is 5' to 3'. The 5' end is phosphate group and 3' end is hydroxyl group. So this is one of the strand, and along with this base phosphate, sugar phosphate backbone, there is a nitrogenous basis on this side. Okay. So this nitrogenous basis, when present in double-stranded DNA, pairs with one another. Now let us see how this basis, which is inside the sugar phosphate bond, how this basis pairs with another nitrogenous base. Okay. So this is the structure of double-stranded DNA or double strand of DNA. You can see the sugar phosphate backbone on both sides. The first strand that I showed you earlier had a polarity of 5 prime to 3 prime. This is having the other strand is having the anti-polarity. You can see it is 3 prime to 5 prime. And different nitrogenous bases are present at the center and they form hydrogen bond with one another. Remember, A always pairs with T, G always pairs with C. A forms double bond with T and G forms triple bond with the C. Okay. So this is how the double strand DNA forms. Okay. So now let's see how the packing of DNA takes place. So DNA, you can see at the start it was a strand. Okay. And this strand coils on one another using a nucleosome. Nucleosome is what? Nucleosome is a structure of histone proteins that is octameric in nature, and it forms or it wraps around it. This wrapped structure is called as nucleosome, which I will be telling you in short while. This nucleosome wraps the DNA around it, and this structure fuses together and creates a bits or a string fashion. Now this structure again thickens up into a loop that is approximately 75,000 base pairs. Now this loop again condenses to form one rosette, which is having 6 different loops. Then again it condenses into one coil that is having 30 different rosettes, and these rosettes combines together. These 30 rosettes of one coil combines together and forms the 2 different sister chromatids. So this is how the DNA is complexly packed into a smaller cell. So now let us see how this histone protein packing takes place. Okay, at the basic level, you can see the bits on a string fashion. This is aligned like this. Okay. So the histone protein is of 4 types. Okay, H1, H2A, H2B, H3, H4. Okay. Out of that, in this structure, I will show you. You can see there are different structures. Okay, so you can see in this structure of nucleosome, this is the histone octamer present at the center. Okay. Out of that, the H1 protein, this is the H1 protein that acts as a linker protein, and other histone octamers that are present in dimeric form. For example, H2A, H2B, H3 and H4. These 4 histones are present in dimeric form and forms an octamer at the center, and DNA is wrapped around this. Okay. So whatever the DNA that is extended to the new nucleosome model or nucleosome is actually called as linker DNA. Okay, and this is the core histone protein. This whole model is called as nucleosome model that is studied. Okay, so now let us see what are the different forms of DNA. There are 3 different forms of DNA: B type, A type and Z type. Now let's see one by one what are the different forms. First of all, let's see the A type. It is a right-handed helix. It is much wider and flattened. Bases are away from the major axis, and it is present in dehydrating condition. Okay, so it is observed in 2 contexts. First, when active site of DNA polymerase is present, or in gram-positive bacteria which is undergoing sporulation. Okay, so A type DNA is present in these kind of conditions. Okay. Now let's see the next one, that is B type of DNA. The most commonly used DNA is of B type. Okay, it is biologically most common DNA. It is right-handed helix that is rotating clockwise or spiral. Okay, then it is having a base pairing of AT and GC. Ideally 10 bases per turn is there, and axes are present at the bases. Okay, in the middle of the bases. Z form is one of the rarest form. It is a left-handed helix. When the salt concentration is more in a biological solution, then only this kind of DNA is shown or seen. There are 12 base pairs per turn in this kind of DNA. Okay. So let us see in one shot the comparison between the DNA. You can see here helix A is right-handed, B is right-handed and Z is left-handed. So Z is one of the DNA helix which is not seen regularly. Okay. Then there are base per turn shown here. Then helical diameter, helical length, shape. Out of that, broadest is the A DNA. Then major groups and minor groups. Okay. Let's see the structure how it looks. You can see the A DNA, B DNA and Z DNA. Okay, A DNA is much broader, whereas Z DNA is much narrower. Okay, so now let us see what is the different effects of different chemicals, acids on the DNA. Okay, so first of all, let's see about acid. From lower than pH 1, if the pH is lower to 1, it will break the phosphodiester bond between the DNA. Okay, and when pH 4 is there, it will result in selective breakage of N-glycosidic bond between sugar and purines. This is called as apurination. Okay, so when the pH is lower, the DNA starts degrading. Okay, under the pH 4. Okay, now when the pH is basic, okay, what is the effect? the base tends to change the polarity. Okay, now regularly you know the DNA is having 5' to 3' polarity and 3' to 5'. This polarity changes above 11 pH. All hydrogen bonds present into the DNA are disrupted. Okay, so but generally the DNA is resistant to basic nature until and unless it is apurinic. Apurinic in nature, or the nitrogen base is removed from it. Then only the base affects the DNA. Okay. Ionic strength. The ionic strength of DNA is negatively in charge because of the phosphate group. Okay, and it helps in dissolution of the DNA into the solution. The GC content is also very important. It tells you about the complexity of the DNA. Higher organisms is having a GC content of 0.5, but lower organisms can have 0.27 to 0.76%. Okay. Solubility. DNA is more soluble than RNA. Then RNA is less stable than DNA because RNA, as I showed you in starting, RNA is having an extra OH group which makes it reactive than DNA. Okay. So these were the chemical properties of the DNA, and this was all about the properties of DNA that we have seen in today's lecture. Thank you so much for attending.